We know that prokaryotes are very simple haploid organisms. They are asexual, which poses a problem because in theory, asexual reproduction doesn't allow for the introduction of new genetic information in the way that sexual reproduction does. In the last videos, we looked at horizontal gene transfer, which includes 4 methods that prokaryotes use to acquire new genes that may help them survive in their environments. We talked about transformation, conjugation, transduction, and transposition. An example of how acquisition of new genes may help a bacterium survive was in the case of antibiotic resistance. So, for example, if a prokaryote picks up a gene for resistance to penicillin, and then it's in an environment where penicillin is present, then that bacterium has an advantage over gram-positive bacteria that don't have the gene and will perish in that environment. Another example was the acquisition of dissimilation plasmids, which allow bacteria like Pseudomonas aeruginosa to break down adhesives and disinfectants that would kill other bacteria. They get an advantage and can live in niches that would be deadly to most other bacteria. In this video, we'll talk about mutation, a universal way that organisms change their genes and develop genetic variation. Your objectives for this video are to define the terms mutation, mutagen, and spontaneous mutation. You should also be able to distinguish between silent, missense, nonsense, and frameshift mutations. You should be able to explain how nucleotide analogs cause mutations and how radiation causes mutations. This is a longer video than normal, so you may want to pause midway, take a quick break, and then rejoin when you feel refreshed. A mutation is simply a change in the genetic material of a cell. And I don't know about you, but when I used to think about mutations, I would think about The Simpsons. And you know how there's this little fish with 3 eyes that's always jumping out of Lake Springfield? That's what would always come to mind. That or frogs that have 3 legs, things like that. But I'd like for you to be a little more advanced in your understanding of mutations. And I'd like for you to know that mutations can be neutral, meaning that they happen. So there's a change in the genetic material of an organism, but it doesn't actually change the way the organism works. And I'll explain that in a little bit. Mutations can also be beneficial, like that antibiotic resistance or the dissimilation plasmids that I talked about. There, cells are acquiring new genetic material, so really they're getting mutations. And in these cases, it's beneficial because the cells can live in the presence of toxic substances. Mutations can also be harmful. Mutations are kind of random. They are changes in genetic material that happen anywhere in any gene. If that mutation happens to occur in a gene, say, that's tied to energy metabolism in the cell, and an important protein gets mutated and becomes non-functional, then that can lead to the death of the cell, which would be a harmful mutation. Mutations can happen a couple of different ways. The first way is a chemical or some environmental condition causes the change in genetic material. This would be an example of a mutagen. So mutagens are chemicals or environmental conditions that cause mutations. Some mutations happen spontaneously. So for example, there's a chemical reaction that happens in a nucleotide base and it converts the base from a cytosine to a thymine or vice versa. Spontaneous mutations happen naturally in the absence of a mutagen. Generally speaking, there are two types of mutations. You have what are called gene level mutations. These types of mutations affect the specific sequence of genes, and remember, genes provide the instructions for making functional molecules in a cell, like protein. So a one nucleotide change, for example, can change the codon for an amino acid, and when that amino acid is changed in a protein, it can totally change how the protein behaves. For example, think about sickle cell anemia. That is the result of a one nucleotide change in a hemoglobin gene. That nucleotide change affects how hemoglobin molecules assemble together. It affects how they orient inside of a red blood cell. It affects the shape of the red blood cell and the ability of that red blood cell to carry oxygen. So it can be, you know, these gene level mutations can be important. You also have what are called chromosomal mutations, and we see those more so in your eukaryotes. Usually these are very significant mutations, and they're usually extremely harmful to the organism that possesses them. Because these chromosomal mutations can so severely affect living organisms, they tend to be a lot more rare than the gene level mutations. For this class, you need to know 4 different gene level mutations. The first is called a base substitution, and this is the simplest type of gene level mutation. Basically, what's happening is we're going to swap one nucleotide in a gene for another. So, for example, maybe an adenine is swapped for a guanine, or a cytosine is swapped for thymine. Here, in a base substitution, only one nucleotide within the gene sequence is changed. So this means that one codon will be affected. Now, just in case you didn't look at the translation videos, let me talk about codons and whatnot really briefly. So as you know, the central dogma of biology says that DNA is the genetic material for all living cells. It's what gets passed on from parent cell to offspring cell. And you know that it encodes all of the information necessary for making the important proteins and RNAs inside of a cell. The next molecule is RNA. RNA is the molecule of protein synthesis. We have to have that in order to make protein. So DNA codes get copied into RNA, and then that process is called transcription, and we've already been through it, so I won't go through that again here. RNA gets converted into or translated into a new language. That's the language of proteins, which uses amino acids as words. The process of going from an RNA language to an amino acid language is called translation. So let's take a look at how this happens. I have a DNA sequence here, and we're going to transcribe it. In order to transcribe it, the first thing you need to know is that there's a protein that does this. It's called RNA polymerase. You might remember that from before. So RNA polymerase. It makes RNA polymers. Wherever this enzyme reads a T, it's going to insert an A opposite. But this is not a DNA base, this is an RNA base. Wherever you see an A, we're going to add what nucleotide? Uracil. Remember, there's no thymine in RNA. You have uracil instead. So wherever we see an A, we're going to put a U opposite. Where we see a C, we're going to put in a G. Okay. Wherever we see a T, again, that's going to be an A. Where there's an A, we'll put in a U because this is RNA, not DNA. Wherever there's a T, we'll put in an A. And we're just going to go down the sequence here. So now we have our RNA sequence. But how do we get to protein? I'm glad you asked. We need a table called the genetic code. So what the genetic code does is it relates RNA bases to amino acids. So the first thing you need to know is that when a ribosome reads an RNA, and these are messenger RNAs, by the way. So when a ribosome reads an RNA, it reads it as a three-letter code called a codon. So we can break this DNA sequence up into three-letter blocks, and each one of these is a codon. Okay, so this is codon 1, codon 2, 3, 4, and 5. Okay, now in the genetic code there are 64 codons listed. 61 are what we call sense codons because they correspond to some amino acid. For example, here we have UUU, and that correlates with phenylalanine. Here we have CCA, and that correlates with proline. Here we have AAA, that correlates with lysine. So again, your genetic code relates codons to amino acids. So what we should be able to do is take the codons from our table and figure out what the amino acids are. And once we figure out the amino acids, then we'll know the sequence that the original DNA encoded. So what we're going to do is we're going to look at the first letter of our codon, which is an A. We're going to find that here on the left side of the table. The next letter of the codon is U. So then after we do the left side of the table, we're going to move across the table from left to right. We're going to find U. So we're going to find A, then move across the A block to find U, which is here. And then while we're in this block, we're going to find the G. So that brings us to this amino acid, AUG, which encodes methionine. So that means that in position 1 over here, we have methionine. And for a lot of organisms, methionine is actually the first amino acid in pretty much every protein. Some proteins will, for some proteins, the methionine gets cleaved off or modified, but for many organisms, that's the first amino acid. So we're going to put a met here for methionine. Next we have AUA. So again, we're going to find A on the left side. Then we're going to move across the table till we find our second A. Oh, sorry. It's another U. Sorry. Let me go back. So we're going to move across the table until we find our U. And then we're going to find the third letter, which is the A. So that's this. That's isoleucine. ILE. So ILE. Okay. Now we've got CGC. We're going to find C on the left side of the table, move across until we find G, which is over here, and then we're going to find our third C. So CGC corresponds to arginine here. Okay. Now, why don't you do the last two? I'll give you a couple of seconds. Okay, so we had U here. We're going to go across the left side of the table, find the U, then go from left to right to find the second U, and then look for the third U. So that's this one here. That's phenylalanine, PHE. And then the last codon was UAA. So we're going to start up here at the U's. We're going to go across till we find A. Then we're going to find A. So UAA is right here. And notice it says stop here. This is one of those nonsense codons. Protein translation stops. It doesn't matter where these stop codons are located. They could be in the first position of the RNA sequence. If there's a stop codon in this first position, then that means the protein doesn't get synthesized. Okay, could happen anywhere. But UAA is a stop, so it means the ribosome is going to let go of the RNA and the protein will not be synthesized any further. Okay, so that's how we go from DNA to RNA to protein. So picking up with the base substitution, we're going to have a DNA sequence, kind of like the one that you see here in purple, okay? But there will be a nucleotide change. And because of that nucleotide change, okay, you can have any number of results. You could have a change in the amino acids that are encoded. You could have a stop codon inserted. You could change the sequence of the RNA, all sorts of things. The first type of mutation, though, is called a silent mutation. A silent mutation. Here, there's going to be a change in one DNA base, but we're not going to change the amino acid that's encoded. Okay, so let me show you this. Okay, so now here is the same DNA sequence from the other slide. All I've done is I've changed one nucleotide and I've highlighted it in red so it's easy to spot. So in our original sequence we had a thymine there, but in our new mutant sequence we have a guanine instead. Okay, let's see if I can remember what the amino acids were. I believe this was a methionine. Then next to that we had isoleucine, which was ILE. Next to that, we had arginine. Next to that was phenylalanine. And then next to that, we had a stop codon. So we've changed one nucleotide. Let's see what happens to the amino acids here. So this is the same. So this is still methionine. We have TAG instead of TAT. So remember that this is going to become AUC. So up here it was AUA. That was the codon. Down here it's AUC. So let's go over and find what AUC is. So we have A, then U, and then C. Look, it's the same amino acid. It's still isoleucine, even though we changed the DNA base. We say that the genetic code has redundancy because more than one codon is assigned to pretty much all amino acids. I think methionine is one of the few exceptions. There's only one sequence for that, but pretty much every other amino acid has at least two codons that it corresponds to. So this means that you can have small changes in the genetic sequence of a gene, but the protein is still the same and it's still functional. It doesn't cause a problem. So this is called a silent mutation because there's no change in the amino acid sequence. In a missense mutation, we're going to have a one nucleotide change, but the amino acid changes. Okay, the amino acid changes. So here's an example. Here's a DNA in purple. Okay, and this is the normal sequence. It's been transcribed here and then translated down here. So you can see that this purple sequence at the top encodes methionine, lysine, phenylalanine, glycine. Okay, and over here where we have our mutant sequence, the cytosine that's located here in our normal sequence has been changed to a thymine. So that's going to change the RNA sequence. So instead of having GGC, instead of having GGC, we have AGC. And AGC corresponds to serine, which is different from the glycine of the original sequence. So there was a change in the amino acid because we changed one nucleotide. That is a missense mutation. Missense mutations can have some pretty significant consequences. If you think about sickle cell anemia, there's a one nucleotide change in hemoglobin B, I believe. And that one nucleotide change changes the shape of the hemoglobin protein. And that shape change translates to a change in how hemoglobin polypeptides come together to make a full protein. And this has implications because those full proteins assemble to form filaments, which distort the shape of red blood cells and reduce the amount of oxygen that they can carry. So missense mutations can be detrimental, but they can also be beneficial. If you think about gram-negative bacteria, they have this outer membrane that serves as a protective border around the cell, right? And the only way that molecules can get to the cell is if they fit through porin proteins. Well, if a porin protein develops a one nucleotide change that changes its shape, then it might exclude certain chemicals like antibiotics from getting into the cell, and that benefits the bacterium. So that was a missense mutation. Let's look at nonsense mutations. In a nonsense mutation, you get a one nucleotide change and it introduces a stop codon. And remember, stop codons stop translation. So that means you get truncated proteins or shortened proteins that may or may not function properly in the cell. Here's an example. So our normal sequence is in purple here, and it's been transcribed to make this messenger RNA, and then that messenger RNA has been translated to give us methionine, lysine, phenylalanine, and glycine as a sequence. When this thymine is mutated to an adenine, we get the introduction of a nonsense codon, that's the AUG. So that was supposed to be AAG, but now it's UAG, and that's a stop codon. So all of these amino acids that happen after the stop codon, they don't get synthesized or added to the protein. We get a truncation. And again, this shortened protein may or may not function in the cell. It might be toxic to the cell. Usually it's not a good thing to have truncated proteins inside of the cell. The last type of mutation is called a frameshift mutation. Frameshift mutations are really interesting. They tend to happen wherever you have a bunch of adenines in the same DNA strand. It turns out that when adenines stack on top of each other, they become very loose and greasy, so that adenines can slip into position or slip out of position during DNA synthesis and transcription. So that results in either the addition of extra adenines or the removal of adenines. And that's what a frameshift is. So in this example, here's our original sequence, our messenger RNA, and then the translated product. Right here, where there will be a phenylalanine eventually, you have a bunch of adenines stacked on top of each other. If one adenine happens to slip out of position during transcription, then instead of getting UUU down here, all of the nucleotides from this point going forward, they move back one space. So instead of getting UUU, we have UUG, and then we have GCU, and then we have AA and whatever comes after. The important thing is we lose the stop codon, and every amino acid after that point where the mutation happens, every amino acid is wrong. So this means that we are likely to get a non-functional protein because these amino acids will not give the protein its proper three-dimensional shape. Now that we've looked at 4 types of mutations, let's look at some mutagens. So remember, mutagens are either environmental conditions or chemicals that cause changes in nucleotide sequence. What you're looking at on this slide are nucleotide analogs. So nucleotide analogs. These are bases that look like one thing, but they act like something else. Here's an example. This is 2-aminopurine nucleoside. And if you compare it to an adenine nucleoside, they look pretty similar. This is our deoxyribose sugar, that's the same. This side of the molecule is the same for both. This part of the molecule, kind of the same. Okay, but if you look at this functional group and this functional group in comparison to the adenine, you can see that they've been reversed. This affects how this base is going to base pair. Okay, to DNA polymerase, the 2-aminopurine looks just like an adenine. Okay, has the same shape, has the same general chemistry. But when it comes to base pairing, the 2-aminopurine does not base pair like an adenine. Which DNA base does adenine usually pair with? Thymine, right? 2-aminopurine doesn't do that. Okay, it doesn't do that. It instead is going to act like a guanine, and it's going to base pair with cytosine. Let's take a look. So I've given you a nucleotide sequence here. This is from some gene, and during DNA synthesis, we've had the incorporation of 2-aminopurine. That's what the A with the asterisk means. So instead of getting a normal adenine, we've got this base getting in. When DNA polymerase comes around and wants to make a copy of this DNA sequence, here's what happens. Instead of pairing with thymine, the adenine pairs with a cytosine, and I've highlighted it in red so that you can see that that's where the change is. Now, you might be thinking, okay, well, that's interesting, and it is, because when DNA polymerase comes and wants to make a copy of this DNA, those cytosines are going to base pair with guanines. So I've highlighted those in red here too. So this means that what should be an AT base pair, it now becomes a CG. And as you saw with the genetic code, changes in one nucleotide can change an amino acid. And changes in amino acid can change the structure of a protein. Remember, the three-dimensional structure of a protein is directly tied to its function. So with changes in structure, you may get a protein that doesn't work the way it's supposed to anymore. And that can be very damaging to a cell or beneficial. It depends upon the circumstance. But I hope what you can see from this example is that our AT becomes a CG. This is 5-bromouracil. 5-bromouracil does basically the same thing. This is an anti-cancer drug. And the way it works is we know that cancer cells tend to reproduce in an uncontrolled manner and at a much faster rate than normal cells. So when you add this to someone's blood and their cells take in this 5-bromouracil nucleoside, it's going to be more likely taken in and incorporated by the cancer cells because they're reproducing DNA so quickly in comparison to normal cells. So what happens is DNA polymerase looks at 5-bromouracil and it looks at thymine. Can't really tell the difference because the sugars are the same. This entire part of the molecule is the same. So for the enzyme, it looks like this is thymine. The only difference is this bromine here. The presence of this bromine changes how this nucleoside will base pair. So that instead of base pairing with adenine, it's going to pair with cytosine. So again, an AT base pair will get changed to a CG. Radiation is another mutagen, and there are two types of radiation that are responsible for causing mutations. The first is called ionizing radiation, and this is very high energy, powerful radiation. We're talking about X-rays, gamma rays, electron beams, things like that. And these high energy waves are able to split water molecules. So remember, this is water. If we split this, we can get hydrogen ions plus hydroxide ions. And those can react with nucleotides, changing their shape, which can change base pairing, and it can change how the polymerases read those nucleotides. Okay, you can also get the formation of hydrogen peroxide, which is H2O2, okay, from this. Generally, ionizing radiation, not a good thing for the cell. Another type of radiation is UV radiation. This is lower energy radiation, and it causes a type of mutation called a thymine dimer. So let me show you what that looks like, and then we'll come back to this. This is the DNA backbone. You can't really see what I'm writing. Let me try yellow. This is the DNA backbone. Okay, here's our deoxyribose. Here's our phosphate. So that's the sugar phosphate backbone. And the two bases that are sitting side by side here, these are thymines. Okay. Now, when UV light hits two adjacent thymines on a DNA strand, they can be cross-linked together. That's what the bold white lines are here. They become attached chemically to each other. They're bonded. They're not supposed to be like this. They come very close together, and it causes the DNA backbone here to buckle. The problem with this is that those DNA polymerases that make new DNA and the RNA polymerases that do transcription, they ride along the backbone of the DNA. And whenever they come to a bulge like this, they stop. So that means that if a cell accumulates a lot of these thymine dimers, a lot of bulges in the backbone of their DNA, and they don't repair that DNA, then DNA synthesis for that cell stops, as does transcription. This leads to death of the cell. This is how those UV light boxes that you might have seen in labs work, or maybe you've seen in like the Brookstone ads, these things that are supposed to sanitize your toothbrush overnight. It's like a little cabinet. You put your toothbrush in, it shines UV light on the toothbrush. This is what they're hoping to accomplish. They're hoping to put enough thymine dimers in the DNA of the bacteria that are on your toothbrush that those cells lose the ability to reproduce and they die. It turns out that a lot of bacteria have repair mechanisms, however, so they can repair thymine dimers. One way that they do this is they have enzymes called photolyases. Photolyases. So photo means light, right? And ly here goes to lysis. Okay, and then the ace on the end, of course, means enzyme. So these are um, enzymes that take energy from UV light and they use that energy to break the bond between the thymines. So it breaks those bold lines from the last figure. And this releases the thymines and the backbone goes back to its normal straight-ish position. The bulges go away. Another type of repair that cells might have is called nucleotide excision repair. Here there's a couple of enzymes that actually cut out the thymine dimer. And then DNA polymerases come in and patch the hole that was made by the missing thymines. Okay, and so they repair that way. So with UV light as a disinfectant, it's really a race against the clock to see whether the cells can accumulate so much damage that they can't repair it, or will they repair all the damage before they die. This was a meaty video. Let's see what you remember. Which end of DNA has a free phosphate group? Is that the 5' end, the 3' end, or what? Right, the 5' end. Remember the 3' end has the free hydroxyl, and the reason why this is important is that DNA strands run anti-parallel. One strand will run 3' to 5', and its complement will run in the opposite direction, 5' to 3'. You'll want to remember this when you are on writing your practice exam, and I ask you to look at a DNA sequence and then write the complementary sequence. Or when I ask you to transcribe a DNA sequence. Which of the following are not true of DNA? Number 1, its strands are antiparallel. Number 2, it's the molecule of heredity. Or number 3, it consists of 4 bases, A, G, C, and U. I knew you'd get that one. Tired, but knowledgeable. The answer here is number 3. It consists of 4 bases, A, G, C, and U. That A, G, C, and U is RNA, right? Not DNA. Which type of mutation results in truncated proteins due to nonsense codons? Is that a silent mutation, a missense mutation, or a nonsense mutation? Right, nonsense mutations. You'll want to be able to distinguish between nonsense, missense, and silent and frameshift mutations. You'll actually have to write some for your exam. In this video, we've talked about mutations, which are changes in the nucleotide sequence of a cell or organism. We said that they can be caused by mutagens, which are chemicals like that 2-aminopurine that we looked at, or environmental conditions like radiation, or these mutations can happen spontaneously when there is no chemical or environmental condition involved. We also distinguish between silent, missense, and nonsense mutations. We also talked about frameshift mutations. Remember, for all of these, we were really talking about base substitutions, where one nucleotide, just one nucleotide in a gene, is swapped out for another nucleotide. And silent, missense, nonsense, and frameshift are all words that describe the effect of swapping that nucleotide out. We talked a little bit about how nucleotide analogs cause mutations. We said that 5-bromouracil and 2-aminopurine cause base pairs that should be adenines and thymines to being cytosines and guanines. And as you know from the genetic code and your study of mutations, that may or may not affect the proteins that are produced as a result. Finally, we talked about radiation. We said that ionizing radiation that comes from very high, powerful electromagnetic waves, we said that it can cause ions to be formed when water molecules are split. And then those ions can attach to nucleotides and change their shape. This shape change can affect the interaction between polymerases and the nucleotides, as well as affect the ability of nucleotides to bond to each other. We also talked about non-ionizing radiation, like UV light. It doesn't split water molecules and form ions, but instead it causes thymines and other pyrimidines, by the way, to form dimers, where the nitrogen-containing bases become covalently linked. They become bonded together. And this distorts the backbone of the DNA and can cause enzymes like DNA polymerase and RNA polymerase to stop functioning. This can lead to the death of a cell. So I know this was a lot of information, and it may take a little bit of time to work through it, but you should have a pretty good understanding of mutations and their implications when you're done. Make sure that you try the practice problems in the practice exam number 2. They are directly related to the material from this lecture, and this material will show up on your actual exam too. Importantly, the practice problems are very, very similar to what's going to be on the exam. So if you can do the practice problems, you should be able to do them on the exam as well. If you have any questions about the practice problems, have any questions about this lecture, I'm available by text, email, and office hours to answer your questions. Good luck.